Welcome to Dr. CM Edcast, your go-to resource for medical learning. Through discussions, case studies, tutorials, and debates, we help you learn, grow, serve.
You know, inside every single one of your cells, there's a code. It's an unbelievably complex and beautiful blueprint that determines pretty much everything about you. This is the story of that code, the architecture of life itself. This is the story of DNA. But before we even get into what the code says, we have to deal with a mind-boggling puzzle. Okay, get this. If you took the DNA from just one of your cells and stretched it out, it would be about 2 meters long. 2 meters! So how on earth does all of that fit inside a space that's 100 times smaller than a grain of sand? It sounds impossible, right? Well, that's the fundamental challenge of storing biological information. To answer that, we've got to zoom in, way in. We need to start with the smallest possible pieces of the molecule itself to understand how it pulls off this incredible feat of packaging. All right, so the most basic unit is called a nucleotide. You can think of it like a little 3-part component. First, you've got a sugar, which is called deoxyribose. That's where DNA gets its name. Then there's a phosphate group, which kind of acts as the connector. And finally, the really important part, a nitrogenous base. This is the actual letter in the genetic code, and there are 4 of them: adenine, thymine, cytosine, and guanine, or A, T, C, and G. Now, what's really cool is how these little units connect. The sugar of one nucleotide links up to the phosphate group of the next, and this bond is called a phosphodiester bond. When you repeat this over and over, you create a long, continuous chain with a really strong sugar-phosphate backbone. Now, notice this chain has 2 different ends, a 5' end and a 3' end. You can think of it like a one-way street. The cell's machinery can only read this code in one direction. And believe me, that directionality is absolutely crucial for everything that follows. But a single chain is only half the picture. The real genius of DNA structure was figured out in the famous model by Watson and Crick. They described how 2 of these chains come together to form the stable, elegant shape we all know. So the 2 strands of DNA, they don't run in the same direction. Instead, they're what we call anti-parallel. One strand runs in the 5' to 3' direction, and its partner runs the opposite way, from 3' to 5'. The best way to picture it is a 2-lane highway with traffic flowing in opposite directions. This arrangement is fundamental to DNA's stability and how it copies itself. So how do these 2 strands actually hold on to each other? Well, the answer is in the bases. They form the rungs of our ladder, and they follow a really strict but beautiful set of rules. Adenine, or A, will only ever pair up with thymine, T, and guanine, or G, will only ever pair up with cytosine, C. A always pairs with T, and G always pairs with C. This is called complementarity, and it's the secret to life's consistency. Because of this rule, the 2 strands are perfect mirror images. That means if you have one strand, you can perfectly rebuild the other one, which is the whole principle behind how DNA gets copied and repaired. So when you put all of that together, you get the famous double helix. It's a right-handed spiral, kind of like a staircase that you climb by turning clockwise. And its geometry is incredibly precise. Each full turn of this spiral is 3.4 nanometers long and contains exactly 10 base pairs. This consistent structure, with a uniform width of 2 nanometers, is what makes DNA so stable and perfect for protecting our genetic code. All right, so now that we know the basic structure of the DNA strand, let's get back to our first big question. Its crazy length. We've seen the blueprint. Now we need to figure out how it's all filed away. Let's just pause on this number again. 2 meters. An incredible length of molecular thread holding billions of letters of code, and it all belongs to just a single cell. It's truly staggering. The question really becomes one of engineering. How did nature come up with a system to coil and fold and condense this molecule thousands and thousands of times without creating an impossible tangled knot? Well, the solution is a total masterclass in organization. The solution starts with a bunch of proteins called histones. You can think of them as tiny molecular spools. See, DNA has a negative electrical charge, and these histone proteins are positively charged. Opposites attract, right? That natural attraction is the key, and it kicks off the first critical step in this amazing packaging process. The basic unit of this whole packaging system is called the nucleosome. What happens is a stretch of DNA wraps itself almost 2 times around a core of 8 histone proteins. This creates a structure that's famously described as beads on a string, where the DNA is the string and the histone cores are the little beads. This here is the very first level of compaction. Our 2 nanometer DNA helix gets wrapped into these nucleosomes, and that forms a 10 nanometer fiber. Just this one step alone shortens the DNA molecule by about 7 times. Not a bad start. But the cell is just getting warmed up. That beads on a string structure then gets coiled up on itself, kind of like twisting an old-school telephone cord to form an even thicker, more compact 30 nanometer fiber. This step condenses it another 6 times over. From here, the packing really kicks into high gear. The 30 nanometer fiber gets organized into these huge loops that are anchored to a central protein scaffold. Then those loops are compressed and folded again and again until finally they form the dense, compact structure we all recognize as a chromosome. This is the final, fully condensed state, and it's so dense you can actually see it with a light microscope, but only when a cell is getting ready to divide. Now, you might be thinking this whole intricate packaging system is just for storage, but this is a really crucial point. It's not. The way DNA is packed directly controls its function. It's a dynamic system that lets the cell decide which parts of its instruction manual are open for reading and which parts are closed off. So this gives us 2 main states of what we call chromatin. The first is euchromatin. This is loosely packed, open for business chromatin. The genes here are accessible and can be read by the cell. On the other hand, you have heterochromatin, which is very, very tightly packed. This dense state effectively locks the genes away, silencing them. This dynamic packaging is literally how the cell regulates its own instruction manual in real time. So from a simple chemical nucleotide all the way up to a complex chromosome, what we see is a system of absolutely breathtaking precision. It's an architecture that solves an impossible storage problem and, at the same time, controls the flow of genetic information. This perfect structure allows life to flourish, which leaves us with a final and pretty critical question to think about. What happens when just a single letter in this vast, perfectly packaged library gets changed?